Okay, so we're going to talk about regulating the cell cycle. So, the cell cycle, it's when the cell divides and it becomes two different cells. So, what regulates this? Well, that's what we're going to talk about in this video. So, here we are, cycle regulation. So, the cell cycle is regulated by different checkpoints. These checkpoints tell whether or not the cell should continue dividing or stop. There's the G1 checkpoint, the G2 checkpoint, and the M checkpoint. So what tells whether or not the checkpoint should keep going or should just stop? Well, that's up to the internal and external signals that the checkpoint receives. These signals can either tell the cell to continue dividing, continue mitosis, and divide into two different cells. Or it could tell the cell to stop, go into this G0 phase and stay stationary, or apoptosis, according to whatever happens. These internal and external signals, I'm going to talk about two of them. One of them is the MPF and the PDGF. These two will tell the cell whether or not to continue dividing or to stop dividing. And we'll talk more about those later. And we'll also talk about what happens when there are disruptions in these checkpoints. When the checkpoints don't work, the signals aren't received, and stuff like that. What happens to the cell when this happens? These are disruptions. What do they cause? Well, in most cases, if the checkpoints are disturbed, and it will cause cancer. Cancer is probably one of the worst things that can happen, but that's what happens when the cell cannot regulate its cycles. So let's continue. Let's take a quick review over mitotic division. So mitotic division is basically when a cell undergoes mitosis and cytokinesis, and it divides into two different cells. These two cells are exact copies of each other. And as you see in the little structure right here, the diagram, interphase is the longest, then followed by mitosis, which is the nuclear division, and then cytokinesis, which is the division of the cytoplasm. So the G1 phase is the first part of interphase. This is where the contents are duplicated besides the chromosome, not the DNA, but the rest of this material inside the cell is duplicated, and the cell continues to grow, and it's getting ready for mitosis. And the S phase is actually where the chromosomes are duplicated. So in humans, all the chromosomes are duplicated. So instead of a 2N cell, it becomes a 4N cell, which is helpful because when it divides into 2, then the daughter cells will all have 2N chromosomes. And then the G2 phase is just double checking everything, making sure everything's duplicated, everything's grown, everything's developed, and then the cell enters into mitosis. During mitosis, prophase, metaphase, anaphase, telophase, when the chromosomes line up in metaphase, divide in anaphase, and the nuclear envelope creates again in telophase. And cytokinesis is when the two cells actually break apart and becomes two different daughter cells. As you can see right here, it's beginning to go into cytokinesis and it's becoming pinched off. The chromosomes have already split and the nucleus is already formed almost, and that will lead to cytokinesis and it'll be two different cells. Alright, so let's talk about the checkpoint. This is what regulates whether or not that cell divides or the cell doesn't divide. So let's talk about the G1 checkpoint. That's the first checkpoint. In the G1 checkpoint, it checks for the different size, the nutrients, the growth factors, and the damage to the DNA. If everything's good, it will continue dividing, and if not, then it'll stop. So if the signals tell the cell to continue dividing, it will move through the S phase and then to the G2 phase to the next checkpoint. But if not, then the cell might be stationary. It might go into the G0 phase. In this phase, it stays stationary and it doesn't divide, and it stays that way. This is the case for some cells in our body, like muscle cells, etc. Now, if we move on to the G2 checkpoint, during this checkpoint, it checks for the last checkpoint before the mitosis actually begins. And once this begins, it checks for the replication. Everything's duplicated, and signals have been received for the mitosis to actually begin. The final checkpoint is the M checkpoint. This occurs during mitosis, during metaphase. This is where they check for the chromosomes attachment to the spindle. If the chromosomes aren't attached to the spindle, then when anaphase occurs, the chromosomes won't be able to move to each opposite end of the daughter cells. So during this phase, that's what it checks for. Until then, the division won't continue. So what causes these checkpoints to either go or stop? Well, that's determined by internal and external factors. So these checkpoints will tell whether or not to go or not. So let's see. The MPF, or the maturation promoting factor, is actually one of the internal signals that a cell receives in order to keep going. And so what is that? The MPF is actually a complex, as seen right here. It's a complex when the cyclin, and the cyclin-dependent kinase come together, and these two proteins come together and join for the complex to be made. So what does it do? It's basically a mitosis promoting factor. So it promotes mitosis to occur. So during the cycle, cyclins are it's a cycle of accumulation and a decrease of cyclin in the cell, and the cyclin-dependent kinase is always around the entire cycle, and it stays there and recycles for every division that occurs. So during the S phase and the G2 phase, the cyclin accumulates. When this happens, the CDK and the cyclin join, and they become the complex MPF. So MPF during the G2 checkpoint promotes mitosis to occur. So when there's an accumulation of MPFs, then G2 will pass the checkpoint and it will give the go-ahead for mitosis to begin. So during mitosis, eventually the cyclin is degraded once again, and once that happens, the degraded cyclin leaves the cycle, and the CDK once again is recycled for the next time. And that tells it when to go and when to stop. So that's an internal signal used by the cells. So what's another signal? This is an external signal, PDGF, or platelet-derived growth factor. So basically, I'm going to give an example as seen as this diagram to what PDGF actually does. So fibroblast cells are actually just cells from tissues in our tissues, and the cells are extracted from the tissues, and in this experiment they're put into a liquid and they're isolated. The cells from the isolated liquid are then placed in petri dishes with different growth mediums. Both have similar growth factors, except this one has the PDGF. So what is the PD? It's a molecule that. So what is it? It's a molecule that attaches to the plasma membrane of the cells. There's receptors on the cell surface that allow the different molecules to attach, and once these attach, they promote and give the go-ahead for the cell to divide. This usually occurs in the G1 checkpoint, and as this occurs, it tells the cell, okay, you're good to go, now go. So when that happens, then the cell divides. So what happens when these don't work? When the checkpoints fail, when signals aren't recognized, what happens? Well, it leads to cancer. So let's just look over this for a little bit. What's a healthy cell? Well, a healthy cell is right here. It's anchorage dependence, which means that it needs a substrate to divide. And a healthy cell will divide until the entire surface is covered, and it'll stop. The density-dependent inhibition will keep it from continuing to divide. And it makes sense. If you take some out, it leaves some space open, and they continue to divide until the space is occupied again. But what about in cancer cells? Well, in cancer cells, that signal isn't reached, that external signal, it doesn't matter to them, and they continue to divide without any factors that limit them, and they continue and continue, and they become almost like on top of each other, and they overcrowd the area. The same thing seen here is seen in this little part of the diagram. These cells are normal, and these are normal, but the cells in here have become cancerous, and they'll continue to divide, and it starts to form a tumor in that area. And this can be very dangerous because they can damage other tissues, and they divide without regulation. The checkpoints aren't regarded in these cancer cells, which leads to abnormal stops and starts. It doesn't stop in G2, it might stop midway through. It doesn't stop at the checkpoints. These checkpoints don't work, and the signals aren't always, the pathways for the signals don't work, and it keeps the cell from dividing, and it might not keep the cell from stop dividing. These cells don't need growth factors. They will continue to grow and continue to grow because they've become cancerous. And that's cell regulation in the cell cycle. And I hope that was helpful.